on the gene at the 220 amino acid position, isoleucine is replaced with valine. Just a little bit of background. SLC6A1 is a gene in humans in the central nervous system that codes for a GABA transporter protein. This protein reuptakes GABA between neuron synapses, and GABA serves as a primary inhibitory neurotransmitter between neurons. When this neurotransmitter is uptaken, it can cause a calming sensation between the neurons, and it basically just stops them from firing. When there's a variation in the protein, this can lead to diseases known as epileptic encephalopathy. Specifically, my patient that I got this variant from is known to have myoclonic atomic epileptic seizures, which can lead to autistic and ataxic features in young children. So they could be autistic, or they could have very limited motor skills. There's only one reported patient have this variant, and there wasn't a lot of information in ClinVar provided on them due to privacy laws, but this variant is known to be a missense variant, and it has an unknown significance in databases such as ClinVar, NOMAD, and TopMed. In this figure, it's a scatter plot showing all of the impact scores of each amino acid, and if you can see in red right there, that's my variant, and it has a very, very low impact score, so it's predicted to not have any kind of a cause on the function or shape of the protein. Specifically, it has a score of 6 on a scale of 0 to 148.5, so very, very low probability. In this figure, it compares the 21 codon window, and my variant has a score of 12 on a scale of 0 to 21, so it has a chance to cause a disease, but it's not one that is most likely to cause any sort of disease in a patient. In this figure, this shows the phenotypic effects of each known variant and unknown variant. In the first column, the purpley color, those are variants from databases such as NOMAD and TopMed that have an unknown significance. And then the second column is benign phenotypic variations in the protein. The next two columns are pathogenic and likely pathogenic, and the yellow column is also variants of unknown significance. If you see the little red line to the right of it, that's my variant, and it falls within the benign phenotypic features. So statistically, it's expected to not cause a change in the protein and not cause epilepsy. But in this figure, it's a box manuscript plot comparing the alpha carbon of each amino acid. And so my variant does not fall within the benign box manuscript. It actually corresponds closely to known pathogenic and likely pathogenic. So this data was taken from NMR data to determine the conservation and stability of the alpha carbon within each amino acid, and with mine falling more closely to corresponding to a likely pathogenic and a pathogenic known variant, this leads me to believe that my variant is more likely to be pathogenic than to be benign, as predicted with the statistical data. These two images are modeled using a program called Yacera, and it's just a 3D model of protein, and this is the wild type. So highlighted in red, you can see where my protein would fall, my variant would fall. It falls on the exterior of the protein, so it's likely to have cellular membrane interactions, and it also falls on alpha helix 8 that is known to be a solute binding domain. So a variation in this alpha helix could cause a significant change in the overall structure of the protein and could lead to disease. To the right of the poster, there's also my membrane modeled within a cell membrane, and you can't really see it from far away, but my variant is also labeled in red, and it shows to be really, really close to the cellular membrane. And then the final figure over there is the global deviation of SLC6A1. So this shows the carbon alpha stability of each amino acid over a time period. So it shows movement within the protein. And the line in blue that kind of goes up is the wild type protein, so a completely normal protein. And then the purple is my variant protein, which you can see kind of flattens out towards the end of the model. And this means that the protein reaches an equilibrium and could potentially mean that it stops functioning or doesn't reuptake GABA like it's supposed to, so it doesn't reach its conformational change, in which case the protein would not move anymore. So this could also support that my variant leads to the epileptic diseases. All of this data was acquired with the help of Dr. Blair and Dr. Jeremy Prokop, who did all the statistical calculations so that I could create the graph scene. And then Yosara is used from code that's sponsoring us from Hudson Alpha. Despite the low impact score that my variant has, I do believe that it can cause a disease, as shown in the box and whisker plot that compares the carbon alpha stability and the global deviation graph, because it doesn't correspond to known variants that are benign. So this would lead me to believe that it has a similar impact that pathogenic variants would have. Any questions?
Oh, you go ahead.
Well, mine was more about the whole.
Okay. So my question was, so basically in most of the scoring, it showed that it had like not much of a change, but then like when they did the surface model of the protein, they showed the variation was on the cell, on the membrane, which is like more impacting of the function. So I guess my question is, do they not, maybe, my question was, do they take where it is on the protein and count enough? Because like the scores, the other score systems that you had seem to just take the, well, what did they take a measure of the other scoring systems?
Like to calculate the impact?
Yeah, the impact calculation, where it said it was like very benign, but then the other ones it said that it was not so benign.
I'm not exactly sure where the numbers came from. Dr. Prokop did those calculations, but it's from the Z-score and a couple of other, I'm sorry, I don't really.
Yeah, the impact score is basically just how stable that amino acid is across species. So we looked at 225 different species, and if it's the same amino acid in each one, then it's highly stable, and it's more likely to cause an impact if it changes. So that's just looking at that one amino acid. It doesn't look at interactions with the membrane. The 21 amino acid score is looking at the 21 amino acids surrounding that particular variant, and it can be very different. So you can have a low prediction for an amino acid, but the motif that it's in can have a major impact. So from the early statistics, we would have said, no, this is not a variant that causes problems, which she came up with. I mean, she figured this out in class. But then we know that the patient has the condition. We know that the 21 amino acid score is much higher than the low impact score that she had. And there's a lot more variation in the movement of the structures after we did the simulation.
Right. I guess my question was it more apparent after you looked at the cell, the protein shape and the membrane and all that? If that was something that maybe they would be doing more in the future, where they study the location on the protein, the mutations location on the protein?
We've talked about that. She came up with it.
Yeah, I definitely think that with the, variant being really close to the cellular membrane, it could lead to making it more pathogenic, just because, like,
It affects the function.
yeah, and like, it could change the cellular membrane itself, like, it could move the protein, I don't know. I haven't thought too much about it, but it definitely just led me to believe that it would be more pathogenic or disease causing.
I'm sorry if my question was a little strange, I didn't think it through before I said it.